Hello everyone, welcome to today's webinar. I'm Jeff Huber, contributing editor at Chemical and Engineering News, and I will be moderating today's event. This webinar is titled The Center for Rapid Online Analysis of Reactions, an Enabling Tech Facility for the Chemistry Community, and is being sponsored by Unchained Labs. CNEN works with sponsors to identify topics of interest and value to CNEN's audience, and consistent with CNEN's mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look at the Help widget at the bottom of the screen or type your query into the Q&A box. If you are disconnected during the webcast, please log in again according to the instructions you received earlier. You are encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. The questions will be answered at the end of the presentation, and as your moderator, I will be posing as many as time permits. This webcast allows certification of participation. To receive your certificate, please click on the certificate icon to download your copy. Please note that CNEN does not endorse any company, products, or services that may be mentioned in the webinars, and that each webinar will be archived at CNEN Online after the live webcast. The presentation today is being sponsored by Unchained Labs. According to Unchained Labs, the company is, quote, all about helping researchers break free from tools that just don't cut it, unleashing problem-tackling products that make a huge difference in the real science they do every day. That's our mantra, our promise, and we own it, end quote. And during the presentation, we will hear from Dr. Mimi He. Dr. He began her academic career as a lecturer at King's College London in 1998, moving to a senior lectureship at Imperial College London in 2003, where she was subsequently promoted to a readership in 2008 and a professorship in 2016. In 2013, she was elected as a Fellow of the Royal Society of Chemistry and was recognized as an Asian Rising Star by an award from the Federation of Asian Chemical Societies. As the director of the Center for Rapid Online Analysis of Reactions facility at Imperial College, she led a successful bid to host an EPSRC Center for Doctoral Training in Next Generation Synthesis and Reaction Technology in 2018, the graduate program of which aims to educate a critical mass of researchers to respond to future research challenges and opportunities created by the data revolution. And during the presentation today, we will also hear from Dr. Benjamin Dedman. Dr. Dedman is the facility manager of the Center for Rapid Online Analysis of Reactions. He uses his 10 years of experience in reaction automation and reaction analysis to support the facility users throughout their adventures in data-rich chemical synthesis experimentation. And also during today's presentation, we will hear from Dr. Paola Farini. Dr. Farini joined the Center for Rapid Online Analysis of Reactions in 2018 and has since become the resident high-throughput experimentation expert and the, quote, tamer of robotic reactors, end quote. And to start things off, I'm going to hand things over to Joe Barco. Joe is the Senior Director of Marketing at Unchained Labs, and he will give a brief introduction.
Thank you, Jeff, for the kind introduction. My name is Joe Barco. I work with Unchained Labs, and I'm going to briefly walk you through what we do. We've been in existence for about 5 years, but we've been selling this equipment through a company called FreeSlate and Symix before that, so there's about 200 configured systems out in the world that do chemistry workflows similar to what we're going to learn about today. There are two sizes for the configurable systems. First is the Big Kahuna, previously called the FreeSlate or the CM3, which is a larger system with a more customizable end-to-end workflow, and the smaller platform called the Junior, which is more of an operational task-driven instrument. These work the same software. The change differences are purely tied to the size of the instrument deck. You can have multiple Juniors and or Big Kahunas in the same lab and have information passed from one to the other so you can complete operations with the context of what was done on the other systems. Over the years, we've developed and refined a series of tools to enable smaller scale reaction screening and optimization. Some of the key ones are listed here. For preparing reactions, you have capabilities to do solid dispensing, liquid dispensing, including heated reagent transfer, slurry transfer, and precipitate transfer. And you can use the included software to set up reactions, or you can import conditions from a DOE software or other third-party software. For running reactions, you have capabilities to do heated reactions, cooled reactions, multiple stirring options, as well as smaller scale pressure reactions. For analysis, there are options to do built-in analysis for things like pH, visual inspection, gas uptake, but you can also integrate with third-party devices such as HPLC, UPLC, GC mass spec, and you can export data into a third-party software for further analysis. We also have built-in analysis tools. This example is called LEA Analysis, which is an Excel plugin where you can line up your experimental setup from your junior or big kahuna with data from your samples. So you can see side by side your experimental data along with the context of how it was set up. I'll conclude my part of the talk with this slide. As interest increases in virtual chemistry and machine learning, there comes a point where you still need to run experiments. And I think Unchained Labs is very well positioned to support machine learning and AI efforts. We already have the piece to do experimental design. We have the capabilities and hardware to run those experiments efficiently in a small scale. And we have the ability to export and produce data. So those of you who are interested in machine learning and AI, you have your part and expertise, which is to close the loop, but we have the other three pieces ready to assist you. And with that, I'll turn the next part of the presentation over to Dr. Mimi He.
Thank you, Joe. Hi, my name is Mimi He, and I'm a professor of catalysis at Imperial College London, where I'm also the director for the Center for Rapid Online Analysis of Reaction, which we abbreviated to RAW. Today, me and my colleagues will be telling you how we have been deploying the Unchained Junior Reaction platforms in our work. Before we do that, let me provide a little context and background to our motivations in using these reactors. The story began with the Dow Molecule Grand Challenge Network, which was funded by the EPIS-RC here in the UK between 2010 and 2020 to promote research aimed at a step change in our ability to deliver molecules quickly and efficiently. One of the challenges is what we term as the smart laboratory. The aim of this initiative is to examine how the lab of the future could make the acquisition and sharing of high-quality complete reaction and process data a low-overhead part of the synthesis workflow. It also looked at how intelligent automation could allow the chemists to be more productive. On this slide, I will outline some of the challenges associated with the synthesis of a molecule. Very often, a chemist has to make many decisions on how to carry out a reaction. Often the choice of a synthetic route is determined not only by the availability of the starting material, but also of the energy sources employed and the reactors, along with the various parameters which will influence the reaction outcome. And here is the vision of how we may dial a molecule. The ultimate aim is to develop a Google map of chemistry, but the starting and endpoints of a reaction can be defined by the user. By utilizing a large quantity of reliable and perhaps real-time data, the program is able to recommend not only all the possible routes using the resources that are currently available, but also identify the potential bottlenecks of the chemistry. There are a plethora of computer-assisted organic synthesis software that are currently available that offer different algorithms in synthetic route planning. This will require access to large datasets, which are only currently available through reaction databases such as SciFinder reactors. However, the accuracy of these predictions will be dependent on the quality and quantity of the reaction data. One of the key missions of RAW is to address this particular issue. RAW was inaugurated in January 2019 at the new Imperial College White City campus. It is a physical facility open to all synthetic chemists in the UK. It comprises the latest state-of-the-art infrastructure, software, as well as expertise to provide training in the use of automated reactors and analytical equipment to support data-driven chemistry. I shall now pass the presentation over to its facility manager, Ben Dittman, who would tell you what we do.
Thank you, Mimi. Hello, everyone. My name is Ben, and I am the facility manager for RAW. Now, I'm going to talk to you now about some of the current limitations in the reaction data that's currently available. So if you look at a traditional, a standard reaction methodology paper, we usually report substrate scope. And typically what we'll do is we've got two reaction partners which are coming together to form a new molecule. And to assess the substrate scope, we will vary one of those reaction partners. So you go along the row and we vary it a dozen times, and once you start finding some of those that don't work so well, you stop. And then you go back and try the other reaction partner and vary that until you find that it doesn't work so well. And then based on the results of those, you can then draw some kind of conclusions about how your reaction would apply to a wider substrate scope covered by this space. Of course, what's implicit in all of these papers, but very rarely explicitly stated, is that there are additional reactions that are carried out which really don't work very well at all, but don't get reported. Now, traditionally, when you're writing a paper, it's very difficult to get these results into your papers. So historically, this information has been lost. So there's just this general general absence of negative results in the literature. And these negative results, it might seem like not that important, but you can't just train software, your reaction prediction software on positive results. You need to also tell it what doesn't work. So we really need to get better at collecting these, the full results. Of course, the problem goes further. Synthetic chemistry is hard enough as it is. We tend to focus our efforts on substrates which are readily accessible and where we think we've got a good chances of success. So the substrate scope we do is biased, and this is actually a problem that's been highlighted by industrial chemists. Many new reactions that we develop don't get picked up by industry nearly as quickly as we'd like. And the reason cited for this is that the substrate scopes being used by academic chemists don't include enough complexity. And so when an industrial chemist finds an interesting new reaction, they can't have confidence that it's going to work on their much more complex substrate molecules. Okay, another problem with the chemical literature is the abundance of reactions which apparently take 18 hours to complete. Very few reactions actually take 18 hours to reach completion. The reality is that chemists are human beings and we need to go home and sleep at some point. And so if you look at this chart here, reaction conversion versus time, the chemist has collected a single data point after 16 hours and reported that data. But actually the reaction was done after 7 hours, but that information was lost. And this is a common feature of the chemical literature. We only really report one data point rather than reporting multiple time points. And the data would be much more powerful if we could provide multiple time points. The final problem, as we see it, is in how we report synthetic procedures. Now, there are ways that synthetic chemists report our methods. And a trained chemist is able to read someone else's procedure and understand what that means, how they can reproduce that. But this is in no way a procedure which could be interpreted by a machine easily, consistently. So it'd be much more powerful if we were to transition to a model where our synthetic procedures are very descriptive. They were written in a structured way which a machine could understand. So we would like to see much more machine-readable procedures in the literature. So within RAW, we have a variety of tools and techniques to help synthetic chemists collect better data. Now, one of these general themes, which you're going to hear a lot about today, is high-throughput experimentation. So RAW has a selection of unchained junior platforms, which allow us to perform hundreds of experiments in an automated way. Now, we can dispense solids, liquids, we can heat and stir, we can take samples out of reactions. And there's a whole variety of operations that we can do with these platforms. That really provides quite a lot of flexibility for how we think about planning our high-throughput experiments. And then, of course, the kind of data you can get from high-throughput experimentation. If we're doing a reaction optimization, we can start thinking about testing all possible combinations of the variables, a full factorial approach, and really have a comprehensive data set about how that how that reaction actually performs under different conditions. And then going back to our substrate scope example, we can do much more diverse substrate scopes because we have the freedom to use the robot to test hundreds of combinations of substrates. The second general area where RAW has a lot of tools is in this idea of in situ reaction analysis and kinetics. So instead of performing hundreds of experiments, in this case we'd perform very few experiments, but we collect a lot of data on those individual experiments. We put in a lot of in situ analysis probes, things like IR spectroscopy, electrochemistry, calorimetry. We take samples out for offline analysis on HPLC. And then we can collect all this data together and start building a detailed understanding of how that reaction performs, what's in that reaction has changed over time. And of course that gives us good kinetic data, builds reaction understanding, and then this reaction understanding then allows process development in the later stages to be accelerated. Okay. And another thing we have a lot of within RAW is automated synthesis capabilities. So we tend to group these into batch or flow, but we have a variety of reactors that allow us to design a procedure on a computer software and then use the software to control the machine, run the reaction, and collect a lot of data about that process. And this obviously has a lot of tie-ins with the high-throughput experimentation, but also the in situ analysis. And of course, one of the big benefits of the RAW facility is that we also provide supporting expertise to help users in using all this fancy equipment. The RAW team, we're made up of Mimi, who you heard earlier, myself, Paola Farides, our high-throughput specialist, and she'll be talking soon. And we've also recently been joined by Dr. Alan Asana as a research associate. And so we're here to support the users in their experiments, and we provide we use our diverse sets of expertise to really push forward the science that our users do. So, what kind of difference can all this high throughput and automation that we have at RAW make to data sets? Well, if we go back to our earlier substrate scope, where we talked about a simple two-dimensional study, changing one substrate partner and then changing the other reaction partner. Well, now if we think about what you can do if you've got high throughput experimentation, we can actually, instead of just looking at the two dimensions, we can look at the whole space that we were covering there and actually provide the data. And now we can see a much more nuanced picture of which ones worked well, which ones worked moderately, which ones worked less well as well. And this data is very valuable. So we can also start to include a lot more industrially relevant substrates in this scope because we can be a lot more adventurous. And we can also provide a much more comprehensive substrate scope. And the idea is if this becomes the routine way of reporting reaction scope, then new reactions will be picked up much more quickly by industry because you'll be able to have some confidence in what it's going to work on and where it won't work so well. And of course, we go back to our picture of the reaction with a single point of data at the end point. Well, if you imagine instead, if you have automation, you can pull out samples at multiple points during the reaction, then suddenly you get this detailed picture about how the reaction is changing over time. Of course, we do a lot of sampling within RAW. That's one of our major benefits of the automation we have. And then finally, I've already told you today about how we have machines which can automate chemical synthesis. We have reactors which are controlled by a computer. Of course, the next step, the next benefit from that is if you run a reaction on one piece of equipment and that's machine controlled, you can take that same procedure and translate that to another lab, the same type of equipment but in another location, and it should run the same way because it's machines. There's not so much of that human variation involved. So we've got interoperable procedures. So at this point, I think it'd be good for us to talk about one of our recent case studies. At this point here, I'm going to hand over to Dr. Paola Farini. She's our high-throughput specialist, and she's going to tell you about some work she's been doing on the reductive amination catalytic reaction.
We decided to study the catalytic reductive amination reaction where a carbonyl compound, like an aldehyde or a ketone, reacts with an amine to form a secondary or tertiary amine in presence of a catalyst, hydrogen pressure, solvent, and for a certain time. We chose this reaction because it's a very important reaction for many industries, from pharma to polymer, agrochemicals, cosmetics, and so on. And also because it's a very simple reaction with a number of variables that can be optimized. For example, catalyst, hydrogen pressure, solvent, temperature, and so on. We decided to keep the catalyst constant with palladium on carbon, hydrogen pressure at 10 bar, and solvent methanol. And we played around with temperature, time, and substrate composition. For the first test, by reading a bit in literature, we saw many examples of heterogeneously catalyzed reductive amination reactions, and we chose to react benzaldehyde with benzylamine in the presence of palladium on carbon at 10 bar of hydrogen. And to prove a point, we just took a one-time point of this reaction after 12 hours at 70 degrees. And instead of obtaining the desired product, dibenzylamine, we obtain 100% conversion of the starting benzaldehyde, but 0% yield of the final product. And this comes back with what Ben was saying. We need the time point analysis of the reaction to understand what happens during the reaction. Why do we get full conversion, but we don't see the product in our mass detector? So performing kinetic studies or time point analysis of reaction is the general need of the chemistry community, and especially of pressurized reactions. And this leads to a technical challenge, that is how to take controlled samples or sample in a controlled manner pressurized reactions without losing the pressure in the reactor. Luckily, Unchained Lab came up with a solution, that is the optimization sampling reactor unit, or OSR. With the design of a chamber and an antechamber, it allows the needle to be pressurized in the antechamber before reaching the reaction vessel, and therefore it is possible to take controlled samples of a specific volume under pressure during the reaction. And this allowed us to take time to perform a time point study of the reductive amination reaction. In this way, we went on to study the original reaction, so between benzaldehyde and benzylamine, and we saw that taking time points during the reaction, after already 30 minutes, we saw that we reached full conversion. However, when we plot the yield of the final desired product, we saw that we got 100% yield after 30 minutes, and then this yield dropped dramatically. And this is due to product decomposition, because in the same reaction conditions that we are running the reductive amination, we have also cleavage of the carbon-nitrogen bond with formation of the starting benzylamine and toluene that was not detected in the mass detector. Therefore, okay, now we know that our one-time point gave us 0% yield because of product decomposition, and we want now to try to hinder this product decomposition in order to study this reaction better. We went on then to optimize the temperature of the reaction, hoping that running the reaction at lower temperature could prevent the product decomposition. Therefore, we ran the reaction at 60 degrees, taking a quicker time point, and we saw that already after a few minutes of reaction, we reached 100% yield of the product, of the desired product, and at 60 degrees, the product decomposition was partly hindered. When we ran the reaction at even lower temperature, like 40 degrees or even room temperature, we saw that this reaction was still very fast, and the product decomposition was completely hindered. So this led us to conclude that the reaction gives us more than 95% yield within 5 minutes from the starting of the reaction, but also importantly, we can hinder product decomposition. And that's important when we want to study multiple substrates, because we want to study them all at the same time within one reaction vessel that I will show you in a moment. And so we need to have a longer time for the reaction in order to allow all the substrates to react. So as I said, we went on to study screening the substrates. And to do so, we used a deck screening pressure reactor, or DSPR, also from Unchained Lab. This is a 48-well reactor vessel, which allows us to run 48 reactions in 2 milliliter vials under pressure. In this way, we screened 4 different carbonyl compounds, 2 aldehydes and 2 ketones, and 6 different amines and all the combination. This allows us to study 24 different combinations, having a 48 well reactor. We could also study these combinations in duplicate to have an average of how the reaction went. We ran all these reactions at 40 degrees for 1 hour, since we know that now the product decomposition can be hindered. With this substrate screen, we faced the first issue of high throughput, which is product quantification or reaction assessment. We could have run 24 different calibration curves for the products in this case. However, this is unthinkable when we think about screening hundreds of different substrates. That gives us hundreds of different products. Therefore, we should come up with a different way to assess the reactions. Our solution in this case was to use the ratio of the area given by the mass detector from our HPLC of the product, of the final desired product, against the area of the starting amine. The starting amine is always in excess in our reactions, therefore it's always present even for full conversion reactions. And the ratio of the area gives us an idea of first, how well the reaction went, and second, also how selective the reaction went. So the highest the number, the better, the higher the number, the better the reaction went, with the highest selectivity towards the desired product. What we can see here is that among these different substrates that we screened, there are some that reacted better than others, as it was expectable. However, we see that we have duplicates reactions running column 1 to 4 are the same as the ones running in columns 5 to 8, all in the same plate. But we can see that the results are not really the same. If we compare, for example, reaction A1 with A5, they should give the same number. They are the same reaction. However, the numbers are quite different. Same goes for A2 with A6, and so on. And this suggests that there is not a good reproducibility across the plate. This is even more clear when we plot the percent deviation between the duplicate reaction across the plate. We can see that there is generally a very large deviation. Most of the reaction give a deviation above 10 or even 30%. Therefore, we have a problem of across-plate reproducibility, which we didn't think about before, and which we should assess before we can go on doing other reactions in the plate. So we came up with a list of possible causes of variation across plate and a possible test or solution for these causes. The first that came to our mind was uneven heating, and we checked that by using an external temperature probe to test the temperature in each plate position. And we saw that in our case, the temperature was quite uniform across the whole plate, so this was not the issue. Another problem can be uneven gas concentration. If the headspace has not been flushed well enough, since the gas enters on one side of the reactor and comes out on the other, there can be uneven gas concentration in the headspace, leading to uneven reactant concentration then during reaction. And this can be solved by having in place a standard protocol for the proper flushing of the reactor. Another issue could be about the steering. We are having an heterogeneously catalyzed reaction where the steering is of paramount importance because of mass transfer. Therefore, it's important to test the steering in various plate positions and ensure to have the right steer bar both in terms of size and shape for the type of steerer that you're using. And finally, if you're using automated liquid dispensing, as we are doing, there can be uneven liquid dispensing across the plate. For example, if you don't have enough starting solution or enough source of the liquid to be dispensed in all positions, the last vials to be filled may be filled with less reactant than the first ones. We checked that. This was also not our problem. So we end up having gas concentration and steering as our possible causes of sources of our variation. And we went on to test those two. To test it, we decided to run a reaction between acetophenone and benzylamine, because we know that this reaction leads to roughly 50% conversion. It's a much slower reaction than the benzaldehyde conversion. Therefore, we could see better the variation between different positions. And we ran the first reaction, flushing the reactor 3 times with hydrogen before final pressurization, and using the small steer bar as we did for the previous substrate screening. And we saw that in this condition, we have a large variety of conversion across the plate. The same reaction gave us conversions between 25 and 75%. And especially we can see from the graph on the right that particularly rows D, E, and F are the ones that gave us the highest variation of conversion of the acetophenone. So we went on to check if it may be a problem of gas concentration in the headspace. To do so, we repeated the reaction by flushing the reactor 10 times before pressurization by keeping all the other parameters the same. And we saw that in general, we managed to improve a bit the conversion. However, the variation still is quite big, especially again in rows D, E, and F. Therefore, we concluded that flushing having a better protocol for flushing the reactor was not the main solution or the best solution for our issue. Therefore, we went on to check the steering. In this case, we changed the steer bar for a fin type steer bar, that is a triangular steer bar, much larger, that can shake better, especially in the vertical axis of the vial. And we can see that in this case, we have slightly lower conversion compared to before, but all these conversions are much more consistent, and except for a couple of outliers, in general, we have conversions between about 40% plus minus 5%. So we concluded that our main issue was due to the steering and therefore to the mass transfer during reaction. And with this new protocol, we can now go back and retest our substrate screening. So we tested again the same reaction as we did before. We see maybe the reactions did not go as well as previously. However, we can see that now the numbers of duplicate reactions are much closer to each other. If, for example, we check reaction A1 with A5, A2 with A6, and so on. And also, when we plot the percent deviation of this duplicate reaction, we can see that the reproducibility greatly improved just by changing the steer bar. And now almost all of the reaction have a reproducibility or percent deviation below 10%. In this way, we managed to prove that reproducibility across the plate can be an issue, and often we don't really think about it. But if we have the right protocols in place, we can improve that and we can solve those problems too. And now that we have a protocol in place for a good reproducibility, the next step will be to go back and try to improve these reactions. With this slide, I would like to give you a few important considerations that I hope could come out from all our talk today, and let's say our take-home message, especially for people that want to move from a round bottom flask to high throughput and automation chemistry. The first most important thing, in our opinion, is to think about the whole process from the sources of your chemicals to the analysis beforehand. All of this you have to think about it before you even start to plan your reaction. Then what we like to say is that the last things come first. So our first questions are about the final analysis of your product. So before you even start to plan your reaction, we want to know how are you going to analyze the reaction? Are your product detectable? Is a quantification method in place? Do you need to run calibration curves and so on? Once you answer these questions, you can start to think about the sources of your reactants. So in which order do you want to add your reactant? Does it really matter for your reaction? In which state do you want to add them? Are they solid or liquid? Can you prepare stock solution? Especially when you think about going from a round bottom flask to high throughput, you have to think that the volume will be much smaller. So if you can prepare stock solution, is a much better option because you can prepare more diluted solution and be more precise in your dispensing. And this raises the next question, that is, are your chemicals soluble in your reaction solvent or in another solvent that can be removed? Then you think about your reaction. Again, you're going towards a smaller scale. Is your reaction reproducible at small scale? Is it reproducible across the plate or even among different plates? And then you have to think about the workup and what you do after your reaction is done. So what is the final state of your reaction? Is it an homogeneous liquid, a biphasic system, a suspension, and so on? Because all these answers will, from all these answers, the type of workup will depend. And finally, if you're using high throughput, most probably you're using an automated dispensing facility or an automated system, and you have to consider all the capability and how able is your system to dispense and to sample your reaction. So what is the minimum dispense volume and dispense weight? What is the minimum sampling volume if you want to take especially multiple samples during reaction? And in our opinion, these are the questions that you should ask yourself before you start to plan your experiment and to design your experiment in high throughput or in automation. And we hope that this is, if you take home anything, this should be your take-home message. Think about the whole process beforehand. With this, I would like to conclude and to thank all of our collaborators and sponsors and partner, and all of you for your kind attention.
All right. Thanks so much for the presentation. And I want to remind our audience that to please submit any questions you might have right now, or thank you to those who have already submitted questions. We're going to get to those and start answering some. So please feel free to continue sending questions in. The first question I wanted to ask was, I believe, for Dr. Dedman. The question asks, do graduate students and postdocs come to you with projects, or do you work with them to effectively design experiments based on the automation and equipment in ROR?
Yeah, it's kind of a combination of the two. The idea is we're a facility that's here to support other researchers' projects. So they will either come to us with an idea, it might be an idea that's fairly well thought out, and then we help them run those experiments to get the best data for their reactions possible. Or they might come along with a very early stage idea in its infancy, and they want to ask our advice on what kind of tools would be available. So it's really a combination of the two.
The next question is for Dr. Farini. This question asks, for the substrate screening experiments, did you try extending the reaction time from 1 hour If your reaction is not going to completion, are you capturing variations in the slope that would not appear if the reaction had a higher conversion percentage?
Hi, thanks for the question. No, for the moment, we only tested 1 hour because, as I said, we encountered the problem that we didn't take into account mass transfer, or we didn't take it as much into account as we should have. So these are preliminary data. We just came up with the protocol to have a reproducible results across the plate. The next step will be to try to improve these reactions. Of course, depending on the substrates, the reaction may be faster or slower, and so the various substrates may have to be improved separately. But we are still planning how to run these future points. About the slope, of course, we cannot take time points during a reaction with the SPR because it's a closed reactor that is pressurized. So it's impossible to take time points, but we will run reactions for different times, 1 hour, 3 hours, 6 hours, and so on. And analyzing it with a mass spec with a QTOF, we can also detect any side products that we are forming and try to figure out what is actually going on in the reaction, because it's a more complicated reaction than we thought. And from that, we can design a better optimization protocol.
The next question is for Dr. Dedman. Can you speak more about how data is shared or how collaboration is done with companies? Is it defined project by project?
Very much project by project. So our facility, for academic users, we're all about giving academics access to this equipment and then generating the data and then trying to make that data as available as possible. When it comes to industrial researchers, we are flexible and will tailor the way we work to industry. But obviously, the more restrictions there are with what can be done with the data, the more we expect in terms of compensation to factor in against that.
Our next question asks, what was the rationale for only using MEOH 10 bar?
Well, this was just a starting point, so we read a bit in literature what were the common conditions. Often these reactions was run in methanol or in water, so we just chose, okay, let's try methanol just for sake of solubility of everything. But as I said, these are just preliminary results. The next step will be to make a design of experiment to study different, screen different catalysts, solvents, pressure, temperatures, and so on. Well, temperature maybe not as much since we have already enough data, but yeah, try to vary the other parameters and see how this reaction can be optimized and generalized.
Next question asks, how are multiple data points taken at elevated temperatures with a volatile solvent?
So I guess we showed two technologies in this talk. One of them is this reactor from Unchained called the OSR, the optimization sampling reactor. In that case, it's got 8 20 mil stirred reactors, and the really cool thing about this reactor is it's got an antechamber system on it. So mid-reaction, you're able to use the robot to bring the needle along, and it'll go down into the antechamber, seal it off, equalize the pressure, take a sample out, and then do the reverse of all these operations to take that sample out of the reaction while it's still under pressure. And with this kind of setup, you can deal with very volatile solvents, provided you put them under enough gas head pressure to keep them in solution. So it's a really cool technology, and that allows us to get multiple data points. Now, with the DSPR, that screening pressure reactor, there is no capability of doing time point analyses on that because you kind of seal it up and then charge in the gas.
Another question asks, have you tried Raman spectroscopy and analytical data fusion to enhance reaction online analysis?
Yeah, I will answer that one. So we are actively looking at Raman spectroscopy at the moment. We've been running some trials with a a major vendor, and yeah, we're getting some very promising results. And I guess the idea is, sorry, just add on to that. The way we work, we like to combine, multiple analytical techniques together to get that detailed picture. And I guess if you're looking at 2 or 3 orthogonal analytical techniques, it gives you much more confidence in that what you're seeing change in your reaction is related to something really happening. But yes, we are using Raman. We wouldn't say much more about it at this stage.
Next person asks, the speakers have mentioned the use of thermochemical and electrical chemical reactions in ROR. What about the use of photochemical reactions? Can such reactions be performed and monitored in this technique?
Yeah, I'll take that one as well. So we do have photochemical setups now. I don't think we've put this on the website yet, but we have 2 major ways of doing photochemical reactions. We have some 96 LED screening setups that allow us to do the kind of high throughput or medium throughput experiments we're talking about today. We can start doing those with photochemistry. This is quite new for us. And then we also have some flow reactors, some continuous flow reactors that have photochemical capabilities. We've got a bit more experience of using those. So we have a variety of techniques, and we can take photochemical processes, between the different ways of doing them.
Next question asks, have you considered or tried using a shaker for the pressurized plate rather than a stirrer?
Yeah, I can take this one. It has been considered, however, although I showed that the reaction with benzaldehyde works quite fast even at room temperature, this is not true for other combination. For example, when we use a ketone like acetophenone, this reaction is much slower, and therefore it needs to be heated. In the system that we have at the moment, we have shaker, but those are not heated, so we have to make a choice between heated reaction or extremely long reaction at room temperature. That's why. And anyways, sooner or later we would have figured out this issue of the wrong stir for the tumble stir that we have. So it's better that we found it out now, and now we know that we should use a different type of stir bar.
Another question says, how do you handle air-sensitive reagents in a high-throughput manner?
Simple one for that is put your robot in a glove box or a purge box in our case. So we're not set up to do air-sensitive reactions yet. We have the purge box there, but while we've been getting familiar with the system, we've been running it under air. But the capability is there, and that's how you do it. Is you just put the whole thing inside a purge box.
Next person writes in and says, I think that chemical reactions require not only its setup and analysis, but also quenching, purification processes to obtain the desired products. What approaches have you tried for these processes, such as liquid extractions or purification by HPLC or concentration?
I will take that question. So this comes down to, I guess, the philosophy of what we're trying to do. Our goal is to study multiple reactions and get some data on how well those reactions have performed. We appreciate there's a lot of interest out there in trying to do high throughput synthesis for the sake of making libraries. We are looking into it. We have a couple of projects doing some work on it, but it's not our main focus. Our main focus is getting that reaction set up, running the reaction, and then getting to the point where you can get a sample out and have that sample worked up enough that you can analyze it and get that data point. And then you'll throw the products away. The idea is work out, do the study of the reaction space, do the optimization and high throughput, and then if there's interesting results there, you can go and reproduce those on a slightly larger scale and isolate the product that way. That would be our kind of main workflow, the way we'd work. But there's certainly ways of doing liquid-liquid extractions and things. If you get creative, you can do it, but that's not our focus for the moment.
Next question asks, can the setup be modified to monitor gas-vapor phase reactions? And if so, how easy would it be to switch back and forth?
I think that's a bit of a tricky question. I think we'd need to talk to Unchained a bit more about that. Possibly. I guess you could run the reaction with gas phase, just gas in there, and try and sample out the gas. But you'd have to think carefully about how you're going to dispense that sample afterwards. So it would need to. Yeah, it'd be very difficult to do.
Yeah, sorry, this is Joe. I'm going to jump in, and just on the OSR, you do have the option to do gas uptake measurements or monitor by gas uptake as well as time.
Yes, I should have mentioned that, yes. So you could do that aspect of it. But taking the samples out would be more of a, not to say it couldn't be done. You've got a nice platform there, but you'd need to come up with some slightly unusual ways of doing it.
Our next question asks, how do you handle the data management in terms of using multiple analytical instruments which probably run on all different software?
I think I can take that one as well, actually. So we're kind of fortunate, and most of our in-situ analysis techniques are based in the Metatoledo ecosystem. So that's one of the things Metatoledo does very nicely with their iControl and ICIR software, is it does a good job of pulling together these different time course data streams and allowing you to process it in, a few different software packages, but then pull the data between them. So where possible, we do that, and we're able to pull in external instruments into those same software packages. But then, this is an open question about how you might start to do that with, shall we say, more diverse sets of equipment. And that's an open question that I guess we'd like to work with vendors on in the future.
Okay, great. Thank you so much. And that's all the time we have for the presentation. I want to say thanks again to Dr. He, Dr. Dedman, Dr. Farini, to Joe for presenting today. Thank you, participants, for being a great audience. Be sure to check CNEN or CNEN Online for information on the next edition of CNEN webinars. Thank you, On24, for technology and production services for today's webinar. And thank you, Unchained Labs, for the sponsorship that made this webinar possible. For CNEN webinars, I'm Jeff Huber. Hope everyone has a great day.